An antibiotic wave moves into a dense green bacterial colony. On paper, this dose is strong enough to wipe out the entire population, but the killing doesn't sweep smoothly through the cells. It slows, and then it stalls. The standard intuition is simple. If the bacteria are vulnerable to the antibiotic and the drug concentration is high enough, the cells should die. That expectation comes from the way antibiotics are traditionally tested in well-mixed liquid cultures, where drugs, target cells, and nutrients are distributed almost perfectly evenly. But real bacterial populations rarely live in such neat conditions. Inside wounds, mucus, and industrial pipes, bacteria form structured groups. Some cells sit fully exposed near the surface, while others are buried deep beneath layers of their peers. In these layered environments, survival hinges on a strange variable. It depends entirely on whether the bacteria have enough food. Delivering a drug to a location is meaningless if the target bacteria are not in the biological state required for the drug to function. Expose a starving bacterial colony to a heavy dose of antibiotics, and they often survive. But add a small amount of glucose to the environment, and a wave of death follows. The drug itself has no trouble physically penetrating the interior of the colony. The molecules diffuse inward, passing right by the cells. The difference is the food supply. As nutrients move into the population, the bacteria on the outer edges consume them first. By the time that flow reaches the inner layers, the nutrients are completely gone, leaving the interior cells deeply dormant. This starvation acts as a metabolic shield. The drug is present, but it needs active bacterial metabolism as an accomplice to actually execute the kill. To isolate this exact mechanism, a 2026 study in Nature Communications immobilized E. coli cells inside transparent hydrogel matrices, allowing researchers to watch the process unfold live. On one side of the matrix was a reservoir containing both glucose and an antibiotic. As those small molecules diffused into the hydrogel, they moved steadily toward the structured bacterial population. The antibiotic used was fosfomycin, a drug specifically designed to interfere with how bacteria build their cell walls. This diagram shows the molecules diffusing toward the colony. Drugs like fosfomycin require cells to be doing biochemical work, like copying DNA or actively building walls to present a target. As the glucose arrives, the outermost bacteria consume it and engage their metabolism. But the interior cells, deprived of that food, remain asleep. Because they aren't building anything, they present no targets for the drug. The outer cells wake up to eat and instantly become vulnerable. They die, turning from green to magenta, creating a sharp, advancing boundary. But the killing front can only advance as far as the food does. The outer, dying layers still consume glucose as the process unfolds, leaving the inner cells completely inactive. The population's collective nutrient consumption creates an invisible wall that dictates the exact speed and depth of the antibiotic's killing power. The researchers tested this bottleneck by altering the inputs. When we dropped the antibiotic concentration by a factor of 10, the death front behaved exactly the same. However, when they increased the glucose supply tenfold, the magenta death front moved significantly faster and deeper into the population. And when they increased the density of the bacteria tenfold, the collective consumption of the outer layer became so intense that the killing stalled. Over 24 hours, less than a tenth of the dense population died. The true bottleneck limiting treatment is the nutrient-controlled metabolic state that allows cells to be killable once the drug arrives. This creates a strange proposition. If dormant cells are harder to kill, doctors could theoretically feed a bacterial infection while treating it to make the drugs more effective. But reports on the study point to a severe consequence of adding excess nutrients to a microbial population. When the death front sweeps through, a small number of naturally resistant cells often survive. If there is a surplus of food left behind, those survivors immediately consume it, regrowing to refill the cleared space. The biological balance is fragile. Supply too little food, and the interior cells stay safely asleep. Supply too much, and the resistant stragglers are given the exact fuel they need to recover. The physical structure of a colony turns antibiotic treatment into a moving front geographical problem. Controlled hydrogels in a laboratory are one thing. The reality of a human infection is far messier. A wound is a chaotic landscape of complex tissue architecture, uneven blood flow, and mixed communities. Clinicians can easily measure the bulk concentration of a prescribed drug, and they can test for known resistance genes. But mapping the microscopic nutrient landscape deep inside a patient's wound is currently impossible. When an infection survives treatment, it often begins as a temporary metabolic survivor, hiding in a starved, low-activity pocket. While this study does not provide a universal dosing strategy, it changes the perspective on how bacterial populations recover from exposure. The goal is shifting toward the ability to map and manipulate the hidden biological conditions that decide if a drug actually works.